By way of introducing myself and telling you a little bit about my credentials to talk about this topic, let me tell you that I taught plant propagation for 10 years at Ohio State before I moved to Clemson. And we set up a tissue culture lab at Ohio State, and we were involved in the early stages of using micropropagation as a tool for multiplying nursery crops, specifically woody trees and shrubs. That's the area that our specialty was in pretty much. And then I moved to Clemson, took my tissue culture program with me there, stayed there for 4 years, and then moved here in 1990, brought my tissue culture program here, and I found out that being department head did not allow me enough time in the lab to be able to keep the tissue culture program going. So I was department head for one term, and then I went back to the lab for a little while, and then I fell in love with World Wide Web technology, set up Aggie Horticulture, and the rest they say is history. But I have kept up with goings on in tissue culture. I still have a great interest in tissue culture, still do a little bit of it from time to time, and that's a little bit about what I want to talk to you about. Tissue culture is, the term micropropagation is just one of the terms that's used to describe the process that people know as tissue culture. Tissue culture means that you can take a piece of plant tissue and use it to recreate the whole plant. It's also known as micropropagation. Some people refer to it as cloning, but as Dr. Hartman mentioned, cloning can be achieved in a wide variety of ways, not just in micropropagation. We'll talk a little bit about some of the disadvantages and advantages of the two techniques. It's also called azenic culture. Azenic culture, by definition, means that you're culturing one organism. And it's in tissue culture, that means that you're culturing the plant tissue and you're not culturing bacteria and fungi. So that's why the term azenic culture was used for a while, but that's no longer in favor. Or sterile culture. Tissue cultures, the plant has to be absolutely sterile because if you allow any bacteria or fungi in, they will very quickly overgrow the culture. And if you haven't done that in lab yet, you will, and you will discover the way that bacteria and fungi can overgrow the plant tissue. This is one of the latter plants that we worked with in tissue culture. This is a selection of Mexican buckeye that I was working with Dr. Arnold on. Had a lot of trials and tribulations with this plant. Got it to multiply like crazy, but could never get it to root coming out of tissue culture. So that's one of the problems we'll talk about. So a clone is a plant that is derived from a progenitor plant purely by asexual means. Clones, if you think of the way clones are produced, clones have an indefinite lifespan. So for example, one of the oldest clones around now is the Cabernet Sauvignon grape. And various estimates have the age of Cabernet Sauvignon grape to somewhere between 1,000 and 3,000 years old. And if you think about the way that clones are produced for the nursery industry and for the fruit industry, we basically take a piece of tissue, usually a meristematic piece of tissue, from one plant, and we produce many plants over and over from that. That's all well and good, and tissue culture is a bona fide way of producing clonally propagated plants. We'll talk about some of those ways, but we'll also talk about some of the exceptions to that rule. And this is one of the exceptions to that rule, which is one of the pinwheel flowering chimeral African violets that we worked with in our lab, and I'll tell you a little bit about some of the complexities that introduces to the process. Plants are able to be reproduced in tissue culture because they're totipotent. Totipotency is the underlying principle of propagation from tissue culture, and it basically means that every living cell in a plant that's capable of further division is able to reproduce the entire plant. So in its simplest form, you can take a single cell, which we call a protoplast, and you can multiply those cells and then eventually cause that clump of cells to develop into the whole plant. Every single cell has the genetics, the genetic background to be able to reproduce the whole plant. Now I said every living cell that's capable of further division, and that's an important principle because there are a few types of cells in a plant that are living cells, but they're not capable of further division because they've lost their nucleus, like a sieve tube element in the phloem, for example. They no longer are able to divide because they have lost they've become enucleate, but they're still living. I should tell you a little bit about what this insert means up here. This is a periclinal chimera, African violet, that you'll learn about the chimeral terminology when Dr. Hartman talks about that. But this type of chimera is one that has a genetic layer that does not contain chlorophyll, and it has two genetic layers that produce chlorophyll and produce chloroplast. And as it turns out that one of the genetic layers that produce chloroplast is the epidermal layer, and when we cause plants to develop on the leaves of this particular chimera, those plants come back genetically entirely green. And that is indicative that only the cells in the epidermis contribute to the development of the new plant. So that's an example of the concept of totipotency. Probably on Tuesday, I'll delve into this process a little bit more, the complexities of it, because it really goes along with the lectures that Dr. Hartman's going to give on chimeras and what you need to know about production of chimeras. So the process of tissue culture begins by taking an explant, and in the case of ornamental plants, in the case of all plants really, that explant can come from any different tissue of the plant. For example, I'm illustrating here the apical meristem, leaf cells, lateral bud, stem cells, and root tissue. And successful tissue cultures and successful plant generation have been obtained from all these different sources, depending on the species that you're trying to propagate. And we'll talk about what morphology contributes to that process in different plants. So for example, you will see examples in just a little bit of taking shoot proliferating cultures from the apical meristem, and from leaves. We can regenerate birch trees, we can regenerate sweet gum trees, we can regenerate tomatoes, just a whole lot of different types of plants from leaf tissue. And from lateral buds, you can treat lateral buds in tissue culture as though they were the apical meristem, and they will regenerate multiple shoots. And then stem tissue, depending on the type of plant that you're talking about, begonias, for example, you can take a small 1 millimeter slice through the stem. A whole bunch of other plants, you can take a slice through the stem and regenerate the whole plant. And the same is true with root tissue. But the key is that those tissues have to be sterile, and they have to be grown under sterile conditions. We sterilize the tissue using a dilute solution of Clorox, just like off-the-shelf Clorox bleach. We dilute that and we're able to sterilize the tissue. Some plants are more successfully sterilized than others. Some plants have endophytic bacteria and fungi that causes problems. But for most of the plants that are successfully adapted to tissue culture, we're able to generate sterile cultures fairly easily. I'm going to talk about two different types of multiplication in tissue culture. The first type is called axillary shoot proliferation, and the second type is called shoot organogenesis. And we've done most of our work with axillary shoot proliferation because it gives us the highest degree of fidelity of multiplication. So you sterilize the rapidly growing shoot tip, and that's usually the type of explant that we prefer to use, because the rapidly growing shoot tip is the easiest to sterilize because it hasn't been out in the dirty environment as long. The worst thing to try to sterilize and put in tissue culture is a dormant axillary bud, because axillary buds seem to accumulate every type of bacterial and fungal spore that there is. Once we get the tissue sterilized and rinsed with sterile distilled water, we put it on tissue culture medium containing a combination of growth regulators that stimulate axillary buds to grow. These growth regulators, the principal one that we use is cytokinin, because cytokinin breaks the apical dominance in a manner similar to what you would get if you were pruning the tip of the shoot. These lateral buds develop, and then as the culture progresses, the lateral buds on these lateral buds break, the lateral buds on these lateral buds break, and within 4 to 6 weeks, you have a mass of small shoots, all because the lateral buds on the shoots that were produced did not go dormant, and they broke out and started to grow. So it's a very rapid proliferation process. One of the first plants that we worked with, and one of my favorite plants to work with, is this Halle Jolivet cherry. Halle Jolivet cherry is a dwarf cherry that's used primarily for an ornamental plant. Here you see one growing in the planter box on the Ohio State campus. This actually was the parent plant from which we took the original cultures of our Halle Jolivet cherry. And this is a picture of the Halle Jolivet cherry clump about 12 weeks into culture, after it's been subcultured a couple of times. And you can see that it's just a dense mass of tiny shoots. If we went in and took all the shoots here that we could root and grow on, we could probably get 150 or 200 plants from this one single shoot tip after about 12 weeks. We went through some interesting calculations, and we did a what if. What if we took a single shoot, and what if we tissue cultured it for a year? And the answer to that what if was we could get about 100,000 plants at the end of that year from that single shoot tip. So that is the power of tissue culture. Before we can achieve those results, I will have to tell you that we have to optimize the media, and the media is the recipe for the agar that we grow the tissue on. The media contains the macronutrients, nitrogen, phosphorus, potassium, magnesium, sulfur, et cetera, et cetera. It contains the micronutrients, particularly iron, manganese, cobalt, zinc, all those micronutrients. It contains the B vitamins, typically. It contains a few growth stimulators like myo-inositol. And the big component of the medium is sucrose, or table sugar, because we have to provide a carbon source, since these plants are not really multiplying or not really doing much photosynthesis on their own. And then we solidify the medium if we're going to grow it in solid medium. We'll talk about some new technology that's being developed now to actually grow it in liquid medium that's very efficient. But once we get that recipe developed, the recipe that we use for Halle Jolivet cherry is different than the recipe that we use for blueberries, and that's different than the recipe that we use for potatoes, and that recipe is different from the one that we use for begonias or broccoli or carrots, or so the optimization of the medium means that you determine the optimum recipe for the medium to achieve the results that you're desiring. Here's an example of the way that we start this process. This is called a growth regulator factorial. Growth regulators are chemical substances that mimic the endogenous hormones in the plant. Indolacetic acid is an endogenous hormone. Isopentyladenine is an endogenous hormone. Zeatin is an endogenous hormone. But in the case of the IAA, we don't use indolacetic acid. We use IBA, indolbutyric acid, or we use naphthalene acetic acid, or we use 2,4-D, because those are growth regulators that mimic the action of the plant hormones. Instead of zeatin, because zeatin is very, very expensive, we use that on some plants, but it's very, very expensive. And in the case of Hallie-Jolivet cherry, we use the cytokinin benzyladenine. So the optimization of the medium means that you determine the best concentration of the auxin and the cytokinin growth regulators that you use to get you the desired result. Back. In this case here, we're using a very low concentration of the cytokinin, and we tend to get a lot of root formation with or without the auxin. So this actually has a lot more roots on it because it's got a lot more root primordia, and that shows the effect of adding the auxin. But it's not necessarily necessary to achieve the results that we're looking for. This is the result that we're looking for right here, or right here, or right here. And that's the multiple shoot formation. So that's what we're trying to do, is multiply these small plants. But you can see that you can go too high with the growth regulators as well. So for every species that you're working on, for every selection that you're working on in tissue culture, you need to do a growth regulator factorial to determine which is the best one for that particular plant. And that's one of the processes that makes the tissue culture fairly complex. And we'll talk about what type of skills the people that are running the tissue culture lab need to have. Contamination is the bane of tissue culture. This is the typical type of culture that you get if it's contaminated with a fungus. Contamination can happen initially from not getting the culture sterile, not getting the piece of explant sterile. It can happen at any point in the process. So that when you're doing the subcultures, if you happen to introduce a fungal or bacterial spore, you're going to ruin the whole culture. I was working with a lab in Northeast Ohio, a nursery lab that was producing rhododendrons. And one time they didn't leave their freshly prepared media long enough to see whether or not it was truly sterile. And it turns out they transferred a lot of their cultures into media that was subsequently contaminated, and that was a real mess. Because every time you throw away a vessel like this, you're probably throwing away potentially 50 to 100 plants that you could have grown on and sold. So it's a very, very expensive process. We'll talk about the equipment and techniques that you need to use to be able to prevent that. Here's an example of that rhododendron lab, but these cultures happen to be sterile, and this is what you're looking for. So each one of these vessels is about 4 inches square, and each one of these vessels contain probably 50 to 100 cuttings that can be rooted. And so you can see in a shelf, lighted shelf in a tissue culture lab, how many plants you can produce. This was the first woody plant that we had a great deal of success with. This is Amelanchier leavis, or Juneberry. Not very common in this area. It does grow in this area, but it's much more common up north. There are fruiting clones of Juneberry that can be used. They produce a fruit that looks a little bit like a blueberry, but they're really very closely related to apple. And these cultures actually were started in my plant propagation class. So we got the hint at how to get the plants to multiply when one of the students was using that. I used to tell the students, well, when we had propagation, go out and find the plant that you want to propagate. And somebody came back with Amelanchier, and it turned out to be a real hit. These are rooted microcuttings. You can see down here a little bit, these are the original leaves that were produced in vitro in tissue culture. You can see how much smaller they are. After we root the plants and then we grow them on in the greenhouse, they turn back into normal looking plants. And then this is a field of the Amelanchier. One of the things I wanted to point out here is how uniform they are. These are absolute clones, and it turns out that they're very uniform, and this is a very good technique for producing things like crabapple and Amelanchier and apple rootstocks and things like that. So the advantages of micropropagation include that you can go from one to many propagules very rapidly. So I told you in the case of Hallie-Jolivet cherry, we could go from a single shoot to 100,000 shoots in less than a year. Now, you can't do that with very many other techniques of clonal propagation. You can do that with seed production of some rapidly growing herbaceous plants, but seed production does not yield clonal propagules. So it's a very, if you find an interesting variant, and we'll talk about what an interesting variant would be. If you find an interesting variant that you want to multiply, if you find a disease-resistant plant that you want to multiply, then you can go from one plant to a lot of plants in a very short period of time. Your multiplication is not seasonal. You're working in a controlled environment, an environment where you control the light intensity, the temperature, and you control the medium that they're growing on, absolutely. So once you get the process started, it continues over and over under very controlled conditions. So it's non-seasonal, propagate year-round, and it has the potential for producing disease-free propagules. What do I mean by that? Most plants that have been grown in the outdoor environment for any length of time have viruses and accumulate diseases. Some of the viruses are asymptomatic viruses, but you can bet that every plant out in the outdoor environment has some type of virus in it. Some viruses are asymptomatic. They don't cause any drag on the plant growth. Some viruses are very debilitating. Some viruses are spread by insects like the rose rosette, and that really, rose rosette disease and potato virus X and Y and all those types of things have to be controlled. And you can control once you get a virus-free plant, you can put it in a screen house, keep it away from the vectors, and it will cut down on the viruses. But in vitro is a good way for growing virus-free plants because they're not subject to any vector that can transmit the virus. However, all plants that are produced in tissue culture are not virus-free. And that was one of the early claims of people when they were starting tissue culture. They were producing clean plants because they were coming out of tissue culture. But if you don't take steps to rid the plant, the initial explant of viruses, then you could potentially be propagating the virus along with the plant. So don't be fooled in believing that all tissue cultured plants are virus-free. Clonal propagation is still the bread and butter of the ornamental plant industry and the fruit industry, as it were. except for certain situations. This is the rose propagation house at Antique Rose Emporium. It's easy to take cuttings off of the roses when they're pruning them, and then they root very readily, and then they grow on very nicely. So that's where tissue culture is not going to replace traditional propagation methods for a lot of plants. Grafting has different advantages. There are a lot of things that you can do in grafting that you can't do in tissue culture, or very easily in tissue culture. So for example, all commercial apples now are produced on dwarfing rootstocks. The dwarfing rootstocks provides a feature to the growth of the fruit. The trees are smaller, they're more prolific, they produce larger size fruit. So, and you can do micrografting in tissue culture. As a matter of fact, we have one lab in South Texas that's micrografting citrus to take advantage of the different rootstock from the clonal scion, but it's not very common, and it's quite a bit more labor intensive. Budding is a very convenient way of clonal propagation. So this is a division of rhizomes of clivia. So because this type morphology lends itself very well to divisions. So in traditional conventional propagation, your equipment costs are quite a bit less than in tissue culture. Tissue culture, you have to have a lab. You have to have an autoclave to sterilize the medium. You have to have a lot of sophisticated equipment to do your sterile transfers. Whereas this mist system up here is basically pressurized water coming through these spray nozzles. I'm sure they'll be doing some mist propagation. So you'll be doing a lot of that, and for the plants that are adapted to this, that's a very easy way to do it. And you don't have to have, it's not rocket science to be able to root a cutting. It's not rocket science to be able to do tissue culture, but you do have to have some pretty sophisticated training, especially to be able to produce the recipes and get the tissue started. We'll talk a little bit about that in a minute. The costs per unit are minimal for this type of propagation. But in micropropagation, the cost can be very low the more propagules that you produce. So, but I don't think you can ever produce a tissue culture explant or a plant through tissue culture quite as cheaply as you can a rooted cutting. But every time you root a cutting, you've got to have stock plants. So you've got to maintain stock plants to give you the cuttings. So if you factor in the cost of holding the stock plants or the advantages of rapid releasing a new cultivar, you have to take all the costs into account when you're comparing the two. Some techniques of traditional propagation will also give you disease-free propagules. For example, there are ways of heat treating and cold treating plants that allow you to produce virus-free plants. And you can keep those growing, like I mentioned, in the screenhouse. So there are ways to do it, but they're not quite as easy as they are in tissue culture. And probably one of the biggest reasons to use grafting is that, well, for some trees, grafting is about the only way you can reproduce a clone. For example, pecans, grafting is about the only way you can reproduce a clone. But a bigger reason is because of the tremendous advantages of rootstocks. We're finding out now, and I didn't think I would ever say this, we're finding out now that it's economically feasible to graft watermelons, to be able to grow watermelons in the field. It's economically feasible to graft tomatoes, to grow tomatoes in the field under certain conditions, or almost all the tomatoes produced in greenhouse are grafted onto rootstocks. So this combination of one genotype growing with the other, the grafting advantage, is very, very important. As I mentioned, the equipment and the facility, I'll show you some pictures in a minute from the tissue culture lab down at Magnolia Gardens and show you some of the intensive equipment that they have. The technical expertise, you don't have to have much technical expertise to do the routine subculturing, but you do have to have some training for that. But the big training comes in the lab manager. The lab manager pretty much needs an advanced degree to be able to deal with all the chemistry involved in optimizing the tissue culture medium. So that's, there needs to be some expertise in there, some training at some point in time. Protocols are still not optimized for all species. I mentioned our frustration with Mexican buckeye, and that's not the only species that we've had frustration with. The first woody tree I tried to propagate in tissue culture was a pin oak. It was a beautiful pin oak called a sovereign pin oak that had all upright branching instead of the drooping lower branches, just a gorgeous tree. And it was propagated traditionally by grafting, except what they found out was that after about 20 years of being grafted, it had an incompatible graft union. And so they'd had all these trees that were just blowing over, just terrible damage that was causing. So we were going to propagate sovereign pin oak, and we beat our head against the wall for about 5 years. Eventually got the plants to multiply, but then failed in the process of rooting it. We were going down that same road with magnolia, but we had a breakthrough in magnolia that allowed us to finally root the magnolia. But in the Mexican buckeye, we had shelves and shelves and shelves of beautifully multiplying cultures. But every time we tried to root them, we got a little bit of rooting, but we couldn't get them to acclimate and grow off in the greenhouse, and that was a problem. Let's see. Another thing, the plants produced in tissue culture may not fit the industry standard. What do I mean by that? Take roses. We produced a lot of roses in tissue culture. We were the original lab that tissue cultured the Belinda's Dream Rose and got a lot of those out to the nurseries to be able to multiply those. We were the lab that produced the Nacogdoches rose or the Grandma's Yellow Rose and got those out. The plants that you get coming out of tissue culture, even after they grow off for a couple of months, they're still fairly small and not really spindly because they're nice plants, but they're not what the industry standard was at the time that we were attempting to do that. The industry standard was a grafted rose onto a very thick, vigorous rootstock, the kind of thing that you see in a packaged roses. They're called packaged roses, where they have a very bulky, thick rootstock. So roses are beginning to be accepted as an industry standard now, but they weren't at the time we were working on them. So you need it needs to fit in with what the industry expects or what there's a market for. What we're finding out is that if you can produce micropropagated plants at a competitive advantage at a price, then the industry standard sort of readapts. And a case in point on that is Nandina. Almost all the Nandina now is produced in tissue culture. And almost all the Nandina produced in tissue culture is done in Magnolia Gardens Tissue Culture Lab, because they've been doing it for 25 years and nobody else knows the secret behind their success. So they start off the plants in tissue culture, but then the young plants, the industry has adapted to the standard of the young plants that are being produced. Depending on the species that you're talking about, the propagules may be fairly expensive. It's hard to produce a tissue culture plant for less than about 65 cents. And for some species, it's really easy to produce a rooted cutting for a lot less than that. So depending on the species, depending on the ease of rooting, there are a lot of factors that need to be considered. Let me briefly review the applications of micropropagation. I think I've told you enough about the rapid buildup of new stock of a superior variety. You discover a new plant, a variegated plant, a disease-resistant plant, a thornless plant, a plant with a different flower color, whatever, and you want to go from one to many very quickly. Micropropagation is the way to do it. If you have an endophytic disease, if you have a virus that you want to clean out of Irish potatoes, tissue culture is the way to do it. If you have a virus in fruit trees, tissue culture is the way to do it. It's better suited to propagation of some species like that were propagated a different way. Do you guys still do air layering on rubber trees, on ficus?
No ficus.
Okay. So and even when I moved down here, I was fairly amazed. I went to a nursery in South Texas, and they were still producing ficus, the Ficus elastica, by air layering. Well, when you're air layering, you have to have a whole greenhouse full of stock plants just to get the plants to air layer. Whereas it turns out ficus is very amenable to tissue culture, and now all the ficus is produced in tissue culture. Date palm. Date palm is very slow to produce offshoots. And so if you have a plant that produces one offshoot every year or something like that, then the amount of space that you have to devote to keeping stock plants is very, very considerable. So date palms is another one that you wouldn't normally think about being produced in tissue culture, but it is. And just tissue culture went big time first in plants that we consider the indoor foliage plants, because they were so easy to produce in culture, and you didn't have to have the tremendous stock plant to go with them. So here, let me show you some of the slides from Magnolia Gardens. We took a trip. I took the nursery management class to Magnolia Gardens last fall, and we toured the nursery as well as the tissue culture lab. Are you going to do a field trip with this bunch this year? Okay. Well, I didn't know whether you were or not. It was sort of, last fall it was sort of dicey because we were trying to mask and trying to go in a small van, and it was kind of a complex process. So here's the media preparation area, and this is probably the amount of media that they will go through maybe in a day. So it's Magnolia Gardens is one of the larger tissue culture labs in the country. They have different types of containers. These are these are containers for their liquid culture. We were asked not to take pictures of their liquid culture because it's still proprietary. But if they get that working, that's going to be very excited, because it's going to drastically reduce the cost associated with propagation. This person has mixed up a big batch of medium, probably Nandina medium, since that's what they do mostly of. And then she's dispensing it into the culture vessels. These are called GA7, polypropylene culture vessels. And then the next step is to put them in these autoclaves. These autoclaves, they have 2 or 3 of these this size. And these autoclaves, this size, run about $250,000 each. So that's one of the expensive parts of setting up a tissue culture lab, but you got to have them. You can have a pressure cooker to get the medium sterilized, but you would have to do 500 loads a day in a pressure cooker, and that's not really feasible. Here's another thing that's very necessary. These are laminar flow hoods that have HEPA filters in them. Supposedly your air in here now is supposed to be HEPA filtered, but a HEPA filter has a very tiny pore size of about 45 microns in the pores. The air is sucked in the bottom and it's blown out through here. So all the air blowing out toward the process, the culture or where the subculture is being done is absolutely sterile. And here are some ladies that are doing transfers, probably again of Nandina. They have 15 or 18 of these laminar flow hoods. These are not that expensive, $10,000 a piece. This is very expensive right here. The labor associated with the person who's trained they are called cutters. For some reason, Magnolia Gardens doesn't let their cutters use headphones or earbuds, but that's one of the, when we set up the big lab at Ohio State, my graduate students had to have their headphones on because it's the drone of the fans and just the process of sitting there doing the cultures. All this has to be done under absolute sterile conditions. If you contaminate your forceps, if you contaminate your culture, then you're going to lose your culture. Here's the picture, a couple of pictures from their growth room. I don't know how many thousands of square feet of growth room that they have, but it's very large, and they've expanded twice since I've been here. These are just standard fluorescent tube that are in this growth room here. But the new growth rooms are being built with LED lights because LED lights don't generate as much heat, and they're very much more adaptable, but they're still pretty expensive. Here's an example of a culture of some type of bulbous plant or lily or iris or something like that, just multiplying like crazy. This culture is ready to be sacrificed to root and grow on in a greenhouse. Now, when you move plants from this stage, the stage 2 or stage 3 multiplying stage, to this stage, you go through a process that's called acclimation. These plants are grown under relatively low light intensity, relatively high humidity, as you can see here. So the first step after they're potted up into flats has to be under very low light intensity and under these humidity temps. And then after about 2 weeks in that, the covers come off. They go to a little bit less dense shade. These again are Nandinas. And then this is probably about 3 weeks after they've come out of the lab. This is about the point that they're ready for sale. So these liners are about ready for sale. And you can just see the scope of the greenhouse that's involved in producing these plants. I think I'll hold on beginning to talk about shoot organogenesis until next time. It's a little bit different pathway. It has a lot of different caveats to it. It's adaptable in some species, but not others. I wanted to save a little bit of time for questions if you have some questions.
Many of them probably don't know what an autoclave is. If you want to explain that to them.
An autoclave is a humongous pressure cooker. In order to get the culture sterile, they have to be heated to 121 degrees centigrade, which is 250 degrees Fahrenheit. And since it's an aqueous medium, In order to get to that temperature, it has to be under pressure. So that's why you have an autoclave. But an autoclave is just a big pressure cooker. I have done tissue culture with a pressure cooker before. And we often get the question about, somebody wants to know, can I do this at home? And the answer is, yes, you can do this at home. There's a person that's been selling a kit to do this called the kitchen culture kit. But if you know what you're doing, you can do it at home, but it's very not advisable because you'll get a lot of contamination.
I have a question.
Yes.
How does the bleach solution not kill the plant?
How does the bleach solution not kill the plant? Most plant tissue that you put in culture has the epidermis intact, and the epidermis is that waxy cuticle. It has a cuticle on it. You'll see when you cut up a piece of tissue, if the Clorox gets inside of the cuticle under the epidermis, it will kill it. And if you use too strong a concentration, it will kill it. If you leave it in too long, it will kill it. So you do a little dance to figure out how you get the sterilization without killing the tissue. So it's a very fine, and there are some other techniques that are used, some other types of compounds, but Clorox is by far the best one to use. Yes?
Same question about the heat. Why does the heat kill it?
The tissue is not put in the autoclave. The medium is put in the autoclave, and you bring it out and you let it cool down. And if you don't let it cool down long enough, it will kill it. And if the air conditioning goes off in your lab, it'll kill a lot of stuff too. So these things are very sensitive. They're not adapted to the heat.
I know you said that like you can do the different like types of micropropagation from all the different parts of the plant. Is it also stuff like cuttings, or do you do it with like individual cells, or is that just like not?
Oh, you've led a segue into the next lecture. I'll talk about the success we've had with working with single cells, and I will show you pictures of the first elm tree that came back from a single cell, because we were the first ones to bring an elm tree back from a single cell. So yes, that can be done. Other questions? All righty then.
We do have sign-in sheets down here, so make sure you visit those on your way out. Let's give Dr. Humber a hand.
Thank you, but that was totally unnecessary.
He is coming out.